It's Muhammad Kashif Munir, the microbiologist. The topic we are going to discuss is the compound microscope or a light microscope. A compound or light microscope is composed of several parts that work together to magnify and illuminate the object being observed. As you know, microscope is used to observe a tiny microorganism under it. It has many parts, and the main parts of microscope include as follows. The first part is eyepiece or ocular lens. The eyepiece is the lens through which the viewer observes the specimen or the object being observed under the microscope. Typically, the power of an eyepiece to magnify the object is 10x or 15x. However, normally the microscopes present in the laboratory contains 10x eyepiece lens. Following eyepiece lens, there are different objective lenses present in the microscope. They may be from 10x, 40x, 100x, etc. Objective lens is the lens closest to the specimen and is responsible for magnifying the image. Microscope typically have several objective lenses of varying magnification that can be rotated into place. As I have already told you that they may be of 10x, 20x, 40x and 100x oil immersion lens. The stage is the platform on which the specimen is placed for the observation. Most of the time, specimens are stick or may be present in the form of smear on a glass slide, and this glass slide contained with smear is placed on this stage. It typically has a clipper or other mechanism to hold the slide in place. The purpose of clips is to hold the slide so that the smear must not move or shake during observation through lenses and eyepiece. Focus knobs are present on the side of microscope. The focus knobs are used to adjust the distance between the objective lens and the specimen to bring the image into focus. Basically, focus knobs are coarse adjustment knobs and fine adjustment knobs. Illuminator is a light source that provides illumination to the specimen. Most of the time, the illuminator is present in the base of the microscope, which throws the light upwards to the objective or the slide placed on the stage and passes through it. In optical microscope, it is typically located below the stage and can be adjusted to control the intensity of the light. Then the diaphragm. The diaphragm is a mechanism that controls the amount and angle of the light that passes through the specimen. We can use the diaphragm to control the intensity of light. Diaphragm is typically located above the illuminator and it can be adjusted to improve the contrast of the image. Sometimes we need a low level of light to see the objective, and at number of times we require a high intensity of light for this purpose. The arm of the microscope is the portion of microscope that connects the base to the eyepiece and objective lenses. It also forms a typical shape of microscope. The base. It is the bottom portion of the microscope. It helps to provide stability and support for the rest of the instrument. That means the instrument rests on the base, which is a wider part of the microscope, the bottom. Other microscope parts may include a condenser lens, which is used to focus the light onto the specimen, and a nosepiece or turret, which holds the objective lenses and allows the viewer to switch between them. Now you can see a labeled diagram of a compound microscope. Almost all parts of microscope are labeled here, and you can ask any question in case of any difficulty. Kindly comment and subscribe the channel. Thanks for watching.